Hello and welcome to another lecture. So this lecture is about seed dormancy and germination. So let's take a look at this figure here from your textbook. This shows you some pre-harvest sprouting of a head in wheat. So this is a wheat head, and you see some pre-harvest or maybe post-harvest, but definitely precocious germination of corn kernels on this corn cob. Right? You can see that all of these are germinating, the little seedlings that are sprouting, even though the corn has not dried up and is not ready to go into the next generation. So in both of these cases, these were mutants that were found due to their strange appearance of germination on the cob or on the head of the wheat before it was time for them to germinate. So the question then was, well, what does normally prevent this from happening? Why don't we see this in more cases than just in these few mutant cases? And so that is what this lecture is about. What is it that is preventing germination at the wrong time and is inducing germination at the right time? So I want to remind you of a few terms when we look at the seeds, and when I talk about certain parts of the seeds, so that you know what each of the parts are. So here on the left you have a kernel of a wheat, and here on the of a wheat plant, and here you have a seed of a bean. So the embryo is down here, so this is all going to be the next generation. Well, this is the maternal generation, the parental generation, right? So what you need to know, this is the radicle, this becomes the root. Over here, this is the radicle. And then you have the leaves, the coleoptile. Remember that that's this little sheet that comes out first that covers the first leaves in grass plants. And then you have the scutellum, which connects the embryo with the endosperm, which is the triploid part of the fruit, or in this case the seed here, that harbors enough storage components, so it could be proteins, but it could also be starch, that can be utilized by the growing embryo. And then in grass plants, there is this alluron layer, and you'll see that becomes important later on. There are certain enzymes that are being made in the alluron layer, and then above that on the outside is the testa and the pericarp. And you see that here as well in the bean. And in the bean, you see that these are the cotyledons. So the cotyledons, this is a dicot, this is a monocot. The actual bean parts, that we eat from a bean, those are the cotyledons. And when you crack them open, you see the little embryo in there with the first leaves and the radicle. So in the case of the beans, the food storage are the cotyledons, while the food storage for the grass plants is the endosperm. And if you look at this in a through microscopy, you look at a microscope slide, you see that this is the alluron layer that I was talking about. So very specialized cells, the endosperm here full of starch granules. Okay, so dormancy is the term that describes a seed that is currently not germinating, and it's not going to germinate until dormancy is broken. Dormancy is induced, and so there are hormones that are putting the seed to sleep after germination, after ripening. And that is called primary dormancy. So the primary dormancy is abscisic acid, that is a hormone that we discussed before in the context of regulating stomata, right? It's a stress hormone as well that regulates drought responses, but it also induces the primary dormancy during the seed ripening process. And then there's something that's called secondary dormancy. And secondary dormancy is induced or kept, so that the seed is kept in a dormant state by environmental factors. So it could be drought, or it could be cold temperatures, stuff like that. And that is still active even after ABA effects wear off. So we have a primary dormancy and a secondary dormancy. They're fluid in their the way which part is which when one is followed by the next. But ABA is important for first inducing the dormancy, and then environmental effects are important for keeping the seed dormant until it's time to germinate. So if you look at this graph here, you see that different species, in this case tobacco, and here brassica, respond differently to environmental influences as well as to endogenous influences that keep the seed dormant. So this is from the website of your textbook. And these little pluses, so this is a plus and here's a minus, that means that it either activates whatever it's pointing to, or that it deactivates whatever it's pointing to, right? So in this case, ABA deactivates radicle growth. So you can see that in tobacco, it's a relatively simple process compared to brassica. This is a rapeseed, where the seed needs to rupture, so the outer shell needs to rupture, that's the testa, and then the radicle grows out. But this is inhibited by ABA, and when that is no longer the case, that is when the seed can germinate. In brassica, it's a lot more complicated. You have an interplay between multiple hormones. So you see that gibberellic acid is involved here, ABA is involved here, light is involved here, regulating gibberellic acid, which then regulates testa rupture. But there's also the gaseous hormone ethylene, and then brasinosteroids that we talked about as well. So all of those have an effect on these various steps that go that go in between a dormant seed and a germinated seed. So this is what we're going to talk about in this lecture today. So germination is inhibited by ABA, and it's promoted by gibberellic acid, by light via phytochrome, by ethylene, and by brasinosteroids. Okay, so your book has this model, and I kind of like this model. It shows that things are fluid, and they're really not like a light switch, toggled on or off, but it's more like a seesaw here, right? It can go a little bit more in this direction, or it can a little bit more in that direction. It can go up and down, and in fact, it can go back and forth a little bit depending on environmental conditions. So let's walk through this and see if we can understand. So we have these three parts. We have perception, what is perceiving it. We have the integration of the signals that are being perceived, and then this is all about the response of the plant. So germinating or not germinating, being dormant or going into germination. So dormancy inducing aspects versus dormancy breaking aspects are the following here. So ABA synthesis. ABA is dormancy inducing. So if we synthesize ABA with this gene, then that will be dormancy inducing. And likewise, gibberellic acid, which I said is important for the germination process, when that is no longer there, or when that can be kept in check, when that is degraded by this enzyme, then gibberellic acid is degraded, and that will also lead to dormancy induction. So you either have no gibberellic acid, but ABA, so therefore we have the blue ABA symbol here, or you can have it the other way around, and that would be dormancy breaking, where you have gibberellic acid synthesis and ABA degradation using these two genes. And so in that case, you would have gibberellic acid. And the tissue itself can also become more gibberellic acid sensitive or less sensitive. So if it's very sensitive, then just small amounts of gibberellic acid will enable it to respond, while if it's not sensitive, then gibberellic acid won't have an effect. So that changes as well. So you can have changes in sensitivity, you can have changes in the amount of ABA, and you can have changes in the amount of gibberellic acid. And so through gibberellic acid and through ABA, you then have signaling. So these molecules then lead to signaling responses downstream of the synthesis of these compounds themselves. And so if you have ABA signaling, then that will allow a seed to go from a non-dormant state to a dormant state. And likewise, if you have gibberellic acid signaling, that will allow a seed to go from a dormant state into a non-dormant state. And this can actually go back and forth. So if these seesaw switch back and forth, then the dormancy state of the seed also can vary. But at some point, when the non-dormant part wins out, that is when gibberellic acid then also can lead to germination. So over time, the seed sensitivity to germination environment becomes greater and greater. So at some point, the seed will simply be ready to germinate. And so just a little bit of ambient environmental changes will be enough then to germinate the seed. And then you have the initiation of the germination process. And so these two compounds, these two hormones are very important in that. And we'll talk about the signaling aspects of that as well. So the bottom line here is that ABA and GA synthesis and breakdown determine the ABA to gibberellic acid ratio, and that favorable environmental conditions at the same time as low ABA-GA ratios then allow germination. And I should say that obviously this is very different. This can be very different from species to species, because species don't all germinate at the same time. They might have different environmental influences that are required for them to germinate. So this is sort of an overview of a general way how seed germination occurs. And of course, it's best studied in Arabidopsis. Okay, so when we talk about signal transduction of ABA and gibberellic acid, we should start by looking at the receptors in the receptor molecules in the cells. So we have looked at receptors before for other hormones, and this one is a pretty different way of signaling, of perceiving the hormone and then using that information for signaling. So we have the left part here where ABA is absent, so no ABA response. And here ABA is present. See, that's the little molecule up here. That is ABA binding to its receptor, and then leading to a number of responses. So we can start over here. No ABA present. We have an unphosphorylated response mediator. So this is the response mediator, and it's not phosphorylated, right? It says dephosphorylated here, and you see that it doesn't have little phosphate groups bound here. So in the unphosphorylated state, it cannot induce the ABA response because it, as a kinase, doesn't have anything to phosphorylate these response regulators that are required for the downstream responses. And the reason for that is because this protein PP2C is binding to this SNRK2 protein, so the response mediator. It's binding and it's preventing the phosphorylation over here. So in the absence of ABA, this guy here represses this guy, no phosphorylation, so therefore no activity downstream here of these other substrates. But when ABA becomes available, then it will bind to the ABA receptor, which will bind to the PP2C protein, which will then relieve the dephosphorylation state. So in other words, it moves up here, it allows phosphorylation. So now phosphorylation occurs. So I say here, ABA binding to receptor relieves dephosphorylation on mediator protein. So the PP2C is no longer active as a phosphatase, meaning picking off phosphate groups. So phosphate groups can now be added. And when they are added, then this enzyme, the kinase here, can transfer the phosphate groups to downstream signaling molecules, which then lead to either gene expression in the case of this BZIP protein or other proteins. These are other substrates of this particular protein. So other responses can occur, other signaling responses that are not necessarily gene expression responses. So that's how the ABA response, signaling response occurs, starting with the ABA receptor, which is a protein, soluble protein. For the gibberellic acid signaling, the process is a lot more similar to processes that we have already discussed. So here we see that this again is a pathway that ends in the degradation through the proteasome. And the proteasome again is activated here by the addition of this ubiquitination tail. So the repressor, in this case, is ubiquitinated by these enzymes, which then take the repressor to the proteasome where the repressor is degraded. Now, how does that happen, and why does that happen? What is this repressor? So it all starts with a set of repressor proteins, and all of these proteins are called DELA proteins. They have a certain set of amino acids, and so that is what makes them similar, and that's what gave them originally this name, that they're DELA repressor proteins. So it's just the name of a class of certain types of these repressor proteins, and they can act differently. So, for example, in this example here, the repressor DELA protein binds to a protein that is a transcription factor that regulates gibberellin regulated genes. So this could bind here, but it can't bind because the repressor protein binds the PIF3-4 protein. If the repressor protein was gone, then PIF3-4 could in fact bind. Now transcription is activated and the genes are expressed. And they are expressed because gibberellic acid has removed this DELA repressor. And the way that it does that is what I just said at the beginning, very briefly, that gibberellic acid binds to its receptor, GID1, and that together with the SLY1 protein then binds to the repressor, adds the ubiquitination tail, takes it to the proteasome, and there the degradation occurs. And so that is why this process is a gibberellic acid induced gene transcription, because it requires gibberellic acid to remove the repressor so that the induction can occur. Okay, so this of course looks very similar to some of the other pathways that we've seen before. And so just not to go over them again, I just wanted you to have them side by side so that you can see that this pathway is very conserved, right? It's always the proteasome. Here we have the ubiquitination through the auxin receptor. Here we have the ubiquitination through the JAS receptor, so the JAS protein that binds to jasmonate. And then here we have that same for the gibberellic acid. So very similar, very conserved overall responses. In all cases, it's about degrading proteins that are repressing a response. Okay, so why do we care about germination? Well, of course, we care about the germination process in general because otherwise plants wouldn't grow. But there's another reason why germination and the steps that are involved in germination are very interesting for certain people. So, malting is a very essential part of the early steps of germination, and the germination of barley specifically. And so if you're interested in beer, or in the process of brewing beer, or how beer gets its taste, then you would be somebody who is studying the germination process in each of the individual steps in barley. So let me just show you some examples here. So here are some barley seeds, and if they are dried, if you were to buy them and you wanted to do some home brewing, if you were to buy them, you get them dried, right? So they are dried down to a water content of about 12%. And then the first thing that happens is, if you are a home brewer, you would have done this, you steep these in hot water. So at that point, you basically imbibe them. Water is soaked up by these grains. And during that time, you get conversion of starch into glucose. So when these seeds are imbibed, they're steeped into water, and this is cold water, right? So that the enzymes, or maybe warm water, so that the enzymes are active, can be active. Now you're at about 45% water content of your grains, and the enzymes now become active, and they convert the starch to glucose. And at that point, the brewer will go back and dry this again. But at this point, you can see the different types of grains here. So either they're roasted a lot or just dried in the kiln in an oven that will then remove the water again. But at the same time as it's removing the water and makes it stable for storage, the glucose and the maltose that was produced here in the malting step is preserved, and the hotter you dry it, so whether you roast it or whether you just dry it, that has an effect on the caramelization, so the way that the sugar reacts with the heat. That's why the amount of caramelization or the amount of drying that you get and roasting that you get of these barley grains has an effect on the taste of the malted product. And of course, malt sugar you can also then derive from these malted grains. Okay, but let's go back to the seed and let's take a look and see how this process occurs. So here's our embryo, and let's say gibberellins are synthesized by the embryo. They are then released to this connection that I pointed out at the very beginning in that slide called the scutellum. They are transported through that and are released into the endosperm. So this is the endosperm, the starchy stuff, starchy part of the grain. And when gibberellins then diffuse into the aleuron layer, that was that one single layer that I showed you at the very beginning, the second slide or so, that is where lots of cell activity occurs, not so much down here, right? This is mostly starch storage. But here you can induce gene expression of certain genes that can break down the starch, such as alpha amylase. So this is a starch breakdown enzyme. And so that is then placed back into the endosperm, so that diffuses into the endosperm, and then it degrades the starch into sugars. And after they're broken down into small molecules, they can be taken up again by the embryo and then transported throughout the embryo and throughout the growing little seedling to allow it to use up the provisions that it has here during its early stages of germination. So this is the aleuron layer, right? This is where the alpha amylase is made, and then it's transported here into the endosperm. Okay, so a little bit more signal transduction so that we can see what exactly happens in those steps that I just outlined for you on a cellular level. So this whole cell here is an aleuron layer cell. Right? So the gibberellic acid diffuses into the aleuron layer cell in steps 1 through 3. So you see steps 1 through 3 here. This is where the gibberellic acid is transferred from the embryo to the aleuron, and then goes into the nucleus. So steps 4 through 6 is where GA then ubiquitinates the gibberellic acid response gene repressor, and then through the repression activates certain genes. So let's look at this step by step. So this is a little bit different from the previous slide because there are different ways how gibberellic acid responds or how it creates the response to gibberellic acid. So in this case, you see the GID receptor. Here's the GA1, so that's the gibberellic acid, binds to the receptor. And then once this occurs, then the DELA protein, that's the repressor, right, that can then be deactivated. So they bind together. This is this whole complex with the E3 ligase that ligates the ubiquitin tail onto the proteins that are then being degraded. So in this case, both of these proteins are being degraded. That now leaves this promoter open, and now you have transcription that can occur from this particular gene. This is called a MIP gene. That's a transcription factor, and that can then act on other genes such as the amylase gene. So this, as a transcription factor, binds to the promoter region of the alpha amylase, makes alpha amylase, and then that gets transported out and into the outside into the endosperm, where it can lead to starch degradation. So that's why steps 7 through 9 here, 7, 8, 9, where the GA induces the amylase transcription, but indirectly because it goes through these multiple steps. And then amylase is transferred out to the endosperm to degrade the starch. Okay. One more aspect that I wanted to add here, because we've talked about this so many times, but we haven't really looked at cellular pathways that lead to the shade avoidance response. So in the shade avoidance response, this now brings together phytochrome that we have discussed in quite a bit of detail and gibberellic acid that we're just now discussing. So I want to show you how that how those two act together in the shade avoidance response. We know that phytochrome is required for signaling, right? For even realizing for the plant that there's shade. So in this case here on the left, you see a cell that is in the sun, and here over on the right, you see a cell that's in the shade. And so if you look at step number 1, high red to far red ratio, so a lot of red light, a little bit of far red light, that is the kind of light that you would see in an open canopy or in a non-shaded region. Right? And the low red to far red ratio, that is where you have more far red. That's where the light comes filtered through the leaves. The green chlorophyll absorbs most of the red light, leaves the far red alone. So therefore now the ratio of the red to far red becomes less than 1, which is characteristic of shade. So that's what you see over on this side. Back to the sun. So if it's sunny, then the phytochrome moves into the nucleus and is activated. So you have the PFR gene form. And phytochrome then can act as a ubiquitinator here of certain proteins, in this case the PIF protein, phytochrome interacting factors. Right? There are different kinds, but there's this one, and when it interacts with the phytochrome, it leads to ubiquitination to the proteasome. Now, PIF can also be present without it being degraded, but if it's present but not degraded, it is bound by a repressor. So this is a DELA repressor. And so therefore, even if PIF is not degraded through phytochrome, then there is still inactivity because of these DELA proteins. So that means just changing the light conditions is not going to allow the PIF protein to become active and act as a transcription factor and lead to transcription of response genes until the DELA protein is removed. And now we know how the DELA protein is removed, right? Because it's a repressor protein that is itself deactivated through gibberellic acid. And so what I write here on the left is that steps 3 and 4, so that is the PIFs are also inactivated by DELAs, so we have no PIF response transcription. But then in the shade over there where we have shading, and we have the low red to far red ratio, the phytochrome is inactive, so it's not going to do this thing. It's not going to take the PIF protein and stick it into the proteasome. And at the same time, gibberellic acid is activated and is binding to its receptor, which then helps the ubiquitination of the DELA protein, liberating the PIF protein, allowing it to bind to its promoters, to the promoter of the places where PIFs can bind, and then turning on genes that have to do with the elongation of the stem. For example, cell wall loosening enzymes that we have discussed in the past as well. So here's an example how you can get phytochrome and gibberellic acid acting together on different parts of signal transduction pathways that together are required for the elicitation of this particular response. And then I have here this, at the end of the lecture, I just wanted to show you, because this is in your book, and it's an interesting aspect, agriculturally speaking, of why it's so important to study shade avoidance responses. So if you look at these corn plants that are very closely, very densely planted, normal plants that are not highly modified through breeding by agriculturalists and horticulturists would not really appreciate this kind of dense planting. So they would show the shade avoidance response that would become long and spindly, and they would just keep growing and growing and growing. But natural variants that are shade tolerant allow much higher density planting. And so through the breeding of shade tolerant species, you can increase the yield on a certain acreage by manyfold. So that's why it's very important to understand what makes shade avoidance and what might be shade tolerance, what it might allow shade tolerance if you're looking to breed certain plants that do or do not express certain types of genes. And that ends our discussion on gibberellic acid and ABA today. See you in class.